This is Bhavani Rajshekar, lecturer from Department of Physiology from SRM Dental College. So today we are going to see about transport through cell membrane. So how the transportation is going to be taken place through your cell membrane. So the transportation is the ionic exchanges from ICF into your ECF or from ECF into your ICF. So these transport mechanism, it is going to be classified into three types: active transport, passive transport and vesicular transport. So first we can see about active transport. So active transport means the ionic exchange which is going to take place with the help of an ATP. So this we call it as your active transport. Passive transport, without the utilization of ATP, the transportation it is going to occur. So this we call it as your passive transport. And the vesicular transport is a special transport where the phagocytosis, endocytosis and pinocytosis it is going to occur here. So first we can see about active transport. As I said already, active transport. So this is going to involve, with the help of an ATP production, the exchange of ions it is going to be taken place. We divide a primary active transport. We divide your active transport into two portions. There is primary active transport and a secondary active transport. So where the primary active transport, the transportation is going to occur with the help of an ATP production. Okay, so this is going to transport the content from one place to the another place. So either from ECF to ICF or ICF to ECF. So this we call it as your pump mediated primary active transport. Secondary active transport is nothing but the ATP which has been utilized already in your primary. So the same ATP will be utilized in your secondary transport mechanism. So that we call it as your secondary active transport. We call it as your carrier mediated transport. So best example for an active transport is a sodium potassium pump. So where the three sodiums they are going to take up with help of a 2 potassium. When 3 sodiums come inside, 2 potassium will go outside, or when 3 sodium goes inside, the 2 potassium will come outside. The first diagram, in this diagram, the first step, the sodium potassium pump binds 3 sodium ions and a molecule of an ATP. So when an ATP has been converted into ADP, okay, so the receptors will receive the content and the gate will open so that the sodium will excrete out. The splitting of ATP provides energy to change the shape of the channel. The sodium ions are driven through the channel. So in the third step, the sodium ions are released to the outside of the membrane and the new shape of the channel allows 2 potassium ions to bind. In the fourth step, you can find the release of phosphate allows the channel to revert to its original form, releasing the potassium ions on the inside of the membrane. So this is the best example for an active transport. So next is calcium pump. Okay, so when a 3 sodium comes inside, the calcium goes outside. The sodium calcium exchanger, this occurs mainly in the plasma membrane. So next is hydrogen potassium pump. So where the hydrogen comes inside, the potassium goes out with the help of an ATP. So next is secondary active transport. As I said already, the already utilized ATP, with the help of that same ATP, the other transportations will occur. So the first step in the diagram shows sodium potassium pump creates an ionic gradient. With the help of the utilization of the same energy, the sodium glucose import transporter loading glucose from your ECF. So the sodium glucose importer transporter releasing glucose to the cytoplasm. With the help of the same energy, so where your sodium potassium transport is occurring, along with that the glucose also transmit. So this we call it as your secondary active transport. This is the best example for your secondary active transport. So next is about passive transport. So passive transport, as I said, the passive transport, there doesn't need any energy. So without the help of an ATP, this transportation will occur. So we divide your passive transport into two forms. So one is diffusion mechanism and the other one is your osmosis. Again, the diffusion, it is going to be divided into simple diffusion and a facilitated diffusion. Simple diffusion, so where the content it is going to move from an higher concentration to one lower concentration. So this is going to move from higher concentration to one lower concentration. So without the help of anybody, so these movements we call it as a simple diffusion. In facilitated diffusion, they need a carrier protein to travel. Okay, so this we call it as a facilitated diffusion. And osmosis, the exchanges will be taken place from higher concentration to a lower concentration through a semi-permeable membrane. We call it as an osmosis. So this is an example for an osmosis where the molecules move from higher to lower concentration with the help of a semi-permeable membrane. We call it as your osmosis. So next is vesicular transport. So vesicular transport, so endocytosis, so phagocytosis will occur. There is any foreign substance which is going to enter into the cell. So first they will think what kind of foreign substance is that. So they will first take up them inside. Okay, then the phagocytosis will be occurring. So the cell eating will be there so that it takes up the content. Then pinocytosis, where the exchanges of the contents will be taken place. After that, finally they excrete them, so which we call it as your endocytosis. After the utilization of everything, they will send the content out. So whatever the waste materials, they will send them out. This we call it as your endocytosis. So exocytosis, where the content it is going to move out. Okay, so where they are going to take up the content and they are going to send out. So next is transcytosis. Endocytosis followed by exocytosis. Transport a substance rapidly through a cell. The HIV crossing a cell layer. Thank you.